Cellular Respiration, an educational video made by Natalie Hablam and Stephanie Powell. So, cellular respiration is essentially a set of metabolic reactions and processes that take place in the cells of organisms to convert energy from nutrients into ATP and then release waste products. I am Natalie, and I will be discussing Krebs cycle and pyruvate oxidization, and my partner Stephanie will be discussing glycolysis and ETC. Enjoy!
Glycolysis is the first stage in cellular respiration. The process harnesses energy by breaking one glucose molecule into two pyruvate within the cytoplasm. The first section of glycolysis is called glucose activation. The first step in glucose activation is energy requiring. This energy is made available when two ATP molecules are hydrolyzed. One ATP attaches a phosphate group to the end of a glucose molecule, creating a glucose 6-phosphate. Glucose 6-phosphate gets rearranged into fructose 6-phosphate, which is an isomer, and the second ATP attaches a phosphate group to the other end of the fructose 6-phosphate molecule. This forms a fructose 1,6-biphosphate. Sugar splitting is the second section in glycolysis. Each ATP transfers a phosphate group to the 6-carbon backbone. The backbone then splits apart to form glycerolaldehyde 3-phosphate, or G3P, and dihydroxyacetone phosphate, or DHAP, which is then converted into G3P. The formation of these two molecules of G3P marks the start of the energy-releasing steps of glycolysis. Oxidization is the third section of glycolysis. Each G3P gives up one hydrogen and 2 electrons to NAD+, reducing it to form NADH. NAD is a reusable coenzyme that functions at the active site of an enzyme. It accepts protons and electrons and transfers them again and becomes NAD+. At the same time, the G3P combines with inorganic phosphate. This results in an unstable molecule called 1,3-biphosphoglycerate, or 1,3-BPG. Formation of ATP is the fourth and final section of glycolysis. 1,3-BPG readily gives up a phosphate group to ADP, forming ATP. This results in a molecule of 3-phosphoglycerate, or 3-PG. As a result, two ATP molecules are formed, one for each G3P. This is called substrate-level phosphorylation, which happens one more time during glycolysis. The molecule of 3-phosphoglycerate rearranges to its structurally different isomer of 2-phosphoglycerate. 2-phosphoglycerate gives up one proton of hydrogen and one hydroxide ion, which combine to form water. The resulting molecule is phosphophenylpyruvate, or PEP. This molecule, PEP, is very unstable and breaks down, releasing enough energy to transfer one phosphate group to ADP, forming another molecule of ATP. This produces a net total of 2 molecules of ATP. At this point, the original glucose molecule has been broken down to 2 pyruvate molecules, each with a 3-carbon backbone.
So when glycolysis has finished, aerobic respiration must still continue. For our second and third stages in cell respiration, the two pyruvate molecules must make their way from the cytoplasm into the mitochondrion. So here is a structure of a mitochondrion. Right here I'm drawing the outer membrane. And then going down here is the inner membrane. The structure that will be referred to most within this section is the matrix here. And then just for another reference, here is the cristae. The second stage in cell respiration, termed pyruvate oxidization, as you can see right here. This begins with the pyruvate molecules entering the matrix, if you think back to the diagram of the mitochondrion. It does this with the helper, a membrane carrier, which is a protein, into the mitochondrion. This passes straight through the outer and inner mitochondrial membranes. Coenzyme A, here, you'll recognize it later in the Krebs cycle. It's also known as CoA, as I'm showing right here, latches itself onto the pyruvate molecule, specifically here, the acetyl group, which now allows the oxidization to occur. During this process, NAD plus is reduced and becomes NADH plus. The carboxyl group left over is then taken out as waste in the form of CO2. The final product is acetyl-CoA. Continuing on in the process, the acetyl-CoA will enter to the Krebs cycle, which you will see next, when energy is needed to continue aerobic or anaerobic respiration. So now the following conclusions can be made about pyruvate oxidization. 2 molecules of NADH are produced for its energy contribution. The third stage in cellular respiration is called the Krebs cycle, also known as the citric acid cycle. It's a series of 8 reactions, as you can see in the picture here, that repeat and The Krebs cycle, also commonly known as the citric acid cycle, is the third stage in cellular respiration. The stage involves 8 reactions that must occur. As you can see, it's a cycle and continues to repeat itself. The reactions work to transfer energy such as ATP, NADH, and FADH2, while CO2 is removed as a waste product. The ratio between glucose molecules that enter glycolysis versus cycles of Krebs is 2:1. If you think back to the picture of the mitochondrion, you will see the matrix where the Krebs cycle occurs. Along the way, enzymes will catalyze, or also known as speed up, the reaction. So, moving on to the breakdown of the actual cycle, acetyl-CoA reacts with oxaloacetate to form citrate. The A has done its job and is no longer needed, so therefore completely removed from the cycle. Now here we have the citrate that was formed. In this stage, the citrate will then change its structure to become isocitrate. This is also known as an isomerization, which kind of goes with the name isocitrate. Kind of makes sense. Here we have isocitrate, which is then oxidized under a redox reaction to form alpha ketoglutarate. As you can see here, an NADA plus ion found from the matrix reduces, to which the electrons are added, creating the energy coenzyme NADH here. So, in this particular stage, alpha ketoglutarate forms succinyl-CoA, and it does this by binding with CoA and an oxidization by NADH, and while all of this is happening, CO2 is released as a waste product once again. So in the fifth step here, the succinyl-CoA becomes succinate by losing its CoA, in which phosphorylations take place for. GDP, or for the long name guanosine diphosphate, becomes GTP, and the phosphate group goes from ADP to create ATP. FAD then oxidizes succinate to form FADH2, another energy coenzyme. Succinate then becomes fumarate. Shortly after, in this stage here, fumarate becomes malate by the addition of water. And the cycle is now ready to repeat again when malate undergoes a redox reaction with NAD+ to produce NADH and oxalate acetate. And in the end, the Krebs cycle then produces overall 6 NADH, 2 ATP, and 2 FADH2.
The electron transport chain is the final stage in cellular respiration. It has 5 major components: complex 1, NADH dehydrogenase; complex 2, succinate dehydrogenase; complex 3, cytochrome BC1 complex; complex 4, cytochrome oxidase; and oxygen as number 5. Proteins are set up in order of increasing electronegativity. The first component is a weak electron acceptor, and the last component is a strong electron acceptor. Electrons are passed from one component to the next. NADH binds to the first component, NADH dehydrogenase. FADH binds to the second component, succinate hydrogenase. Hydrogen ions are pumped into the intermembrane space. The mobile electron carrier, ubiquinone, carries electrons from NADH dehydrogenase to cytochrome BC1. The mobile electron carrier, cytochrome C, carries electrons from cytochrome BC1 to cytochrome oxidase. FADH2 works the same, but it starts at succinate complex. Since electrons from FADH2 pass through fewer electron carriers, it makes less ATP. Each molecule of NADH produces 3 ATP and 3 hydrogen ions. Each molecule of FADH2 makes 2 ATP and 2 hydrogen ions. When the 2 electrons leave cytochrome oxidase, they combine with oxygen, final electron acceptor, and hydrogen ions to form H2O. Oxygen is used because it is the only element with a strong enough electronegativity to strip the last component of the ETC of its electrons. ETC is used to create an electrochemical gradient to power ATP synthesis. Chemiosmosis is the production of ATP in cellular respiration. The movement of ions from an area of high concentration to an area of low concentration through the membrane protein. This results in the release of hydrogen ions. Chemiosmosis can only occur after the electrochemical gradient has been created in ETC. Hydrogen ions are now forced through the special enzyme called ATP synthase embedded in the inner membrane. Hydrogen ions flow from the intermembrane space into the matrix. The flow of hydrogen proteins provides energy to make a bond between ADP and inorganic phosphate. This is how ATP is made via oxidative phosphorylation.
And thank you for watching our video on cellular respiration. I hope you learned lots and you enjoyed it.